Hello, everyone. Thank you so much for joining us today for this amazing talk by Professor Gorbet from University of Waterloo. This is our great honor to have her today in our seminar series. Before starting this talk, I just want to, remind you some of our housekeeping notes. You can ask your question in Ask a Question box in the bottom of your screen, and we have the chat box that if you wanna send a hi or greetings to your friends and colleagues, you can use the chat box. But question, please send it in the Ask a Question box. We have a Mohsen can turn on the poll. Also, you can actually take this survey to help us improve the seminar series. And the next speaker is Professor Gillen Tibbetts from Simon Fraser University. He's talking about investigating the mechanism of hypertrophic cardiomyopathy using human-induced pluripotent stem cell cardiomyocytes. And if you have any question and if you want to get the most updated information about our seminar series, please follow us on Twitter page, hashtag Translational Biomedical Engineering. And also, if you have questions, you can email the organizer, myself, and Mohsen. Last but not least, we always want to thank Montreal Institute that sponsors this seminar series from almost the beginning of this series that we started almost more than. So by that, this is my great pleasure to introduce Professor Gorbet. She's an assistant professor in the Department of System Design Engineering at the University of Waterloo. Trained first as a bioengineer in France, Université de Technologie de Compiègne, in 1993. She completed her PhD in chemical engineering at the University of Toronto in 2001, investigating the mechanism of material-induced thrombosis. Joining Toronto startup Rimon Therapeutics in late 2000, Professor Gorbet, as a senior biomaterial scientist, was responsible for the evaluation of biological efficacy and safety of their wound healing biomaterials and supported preliminary filing with the US FDA. In 2007, her strong interest in investigating mechanism of biocompatibility and in educating the next generation of engineers led her to join University of Waterloo. As a researcher and director of the Materials Interaction with Biological System Laboratory, Dr. Gorbet studied the development of better in vitro models and tools to assess biocompatibility and the study of the mechanism involving material-induced cell activation. Over the past 14 years, she has collaborated with the startup companies, ophthalmic industries, clinicians, scientists, and engineers from Canada and around the world, and has received over $1.5 million in research funding from NSERC, Canadian Institute of Health Research, CIHR, and through industry-sponsored contracts. Professor Gorbet also spearheaded the creation of the Biomedical Engineering undergraduate program, which has become one of the largest direct entry and full co-op undergraduate biomedical engineering program in Canada. In September 2014, she was appointed inaugural biomedical engineering director. From April 2019 until August 2020, she served as interim system design engineering chair. On national and international stages, Professor Gorbet has presented her research and organized conferences and symposia. She has authored 40 papers, 5 book chapters, and more than 60 conference abstracts. Professor Gorbet is a member of the Canadian Biomaterials Society and of the Association for Research Vision and Ophthalmology. She served as the president-elect, president, and past president of the Canadian Biomaterials Society from 2010 to 2013. She was inducted as a member of the International Society for Contact Lenses Lens Research in 2011 and is also a member of the College of Reviewers for CIHR. Professor Gorbet has also been actively in community outreach with the non-profit organization First Canada for inspiration and recognition of science and technology. She has coached or mentored several first team engaging youth from 8-year-old to 17 to learn about science and design, judged at many events, and has also contributed to the organization of first competitions in her community to support and recognize youth achievement. This list goes on and on, but really congratulations for this long list of achievements, Professor Gorbet. By that, I invite you to share your screen, and the floor is yours. Thank you so much.
Thank you so much for the kind introduction. So let me make sure I'm going to share the right screen. And I most likely you see me, but it should work. Just a second. All right. Perfect. I'm assuming you see the talk.
Yes.
All right. Great. Thank you so much. It is really with great pleasure that I'm talking to you today about my research, and it's and I guess before we talked about my research, I would like to acknowledge actually that the University of Waterloo is on the traditional territory of the neutral Anishinaabe and Onansumi people, and my lab actually and my house where I'm talking from actually is situated on the Alderman Tract, which was a land granted to the Six Nations. And so I think it's important to recognize as part of the work we do that part of my teaching and my research is taking place on that land, and I'm hoping I can help with reconciliation. So really today what I wanted to talk to you about is, I know the series around translational medicine, biomedical engineering, and what I was interested in doing is telling you a little bit about how, you know, sometimes your story changed roads depending on what happened in your research. And so I decided to talk a little bit about how from developing in vitro models of ocular biocompatibility, we ended up starting investigating a new cell population that was on the ocular surface and trying to understand their role in the inflammatory response. So you're going to see some of the biomedical engineering in vitro models that we've done, but you're also going to see some of a little bit more of a biology at a higher level, just because I want to make sure I stay on time, but just to introduce you to a new type of cells that are present in the eye, and that could be fairly important actually for biocompatibility. And so I know there's many people on this call, you know, biocompatibility, I believe most of you know what it is, but I think because this is what I do, it's really important that I actually remind everyone that, you know, one of the definition or the most recent definition that really try to really takes into consideration the fact that biomaterials are just not passive material, is that we want to actually have them perform their desired function without eliciting any undesirable local or systemic effect, but also generating the most appropriate beneficial cellular or tissue response in that specific situation. And what I highlighted are actually two things that relate to, you know, the cells that are present, but also the specific situation. And so when we start talking about, and I'm just going to get a pointer, when we start talking about, you know, biocompatibility, we know there's a material and there's physical and chemical properties of that material that are really important. We also know that there's a biological response with the cell material interaction, and there's protein absorption, and there are very different things that will actually affect how the cell will respond. And we also need to think about, you know, the patient or the person in whom we're going to, you know, implant, or with whom that device is going to interact one way or another. And so as part of my lab, what we're interested in when we're looking at biocompatibility is trying to understand it. So are there ways that we can better understanding what's happening? And also, can we test it effectively in vitro? Because that's one of the important part about as we develop more models and as we develop more biomedical devices, we also want to contribute to reducing the use of animal. And so as part of our lab, we also think about as we're talking about biocompatibility testing of medical device is, the 3 R's that you might be aware of: replacement, reduction, and refinement. And really what my lab sits, it's really trying to see if we can answer some of the question around biocompatibility by developing new models in a way. And so when we're looking a little bit about what we do in my lab, which is called Material Interactions with Biological Systems Lab, basically we're trying to integrate various mechanism in biocompatibility. And so we've looked at we've studied the cardiovascular system and looking at cardiovascular device. And we've got we just finished a collaboration with a cardiac surgeon in St. Mary Hospital, looking at bleeding risk with cardiopulmonary bypass. We also have a collaboration with Dr. Tom Willett at the University of Waterloo, where we're working with him in testing the biocompatibility of the 3D printed nanocomposite. And we also, try to answer biocompatibility question that industry-sponsored project bring to us, so looking at various medical device depending on what their interest is. And today, what I'm going to focus on mostly is some of the work we've done around the ocular surface or the ocular system. And so we've looked at different components of the ocular system. We've looked at intraocular lenses, trying to understand a little better what happened with macrophage and lens cells. We're looking at mechanobiology, trying to understand, how corneal epithelial cells respond to shear and some of that, and other mechanical stress. One of the work we're doing is actually with Dr. Yim, looking at keratoconus, where there's significant mechanical stress on the cornea due to eye rubbing, but also to the change in the membrane that the corneal epithelial cells are on, with collagen being weaker. And also what we've been quite interested in is looking at biocompatibility model of the ocular surface. And some of that work, and you're going to see, is actually done with Dr. Lyndon Jones at the University of Waterloo and the Center for Ocular Research and Education. And so what I'm going to focus today mostly is, looking at when we're trying to answer some of those questions around biocompatibility, and really using actually contact lens as an example of a medical device where there are some questions that we have around biocompatibilities that have not been answered, and they're difficult to answer because the ocular system is so complex in a way and so difficult to sample compared to if you're looking, for example, as testing a stent or something. You can take a blood sample fairly easily. It's a little bit more complicated with the ocular system because it's difficult, and there's also a lot fewer, or the concentration of proteins are different. And so it's a little bit more complicated around that area. Something that's really important, and I'm sure most of you know, but again, I just want to reiterate this, is that when we say biocompatibility, it's not quite the same as toxicity. There's a difference between, biocompatibility goes beyond just looking at a cell if it's dead or not. So that's what cytotoxicity is. When we're looking at biocompatibility, we're thinking about, are the cells activated? Are they responding differently? And so that's kind of what we're interested in a way sometimes, and in some of the model we build is really trying to understand what role they play, but also looking at, what do those change in cell activation lead to? And that, it may lead to change in morphology, membrane surface receptor go up and down. That changes adhesion and activation. There's also release of inflammatory mediators that leads to activation. And so whenever we're looking at biocompatibility, we and sometimes we do, live and dead assay and SIGs, but, a lack of cell toxicity does not imply that your material, your sensor, your treatment is biocompatible. And so as we design our test, we try to think about, what are the way we can actually assess that biocompatibility. And that's why sometimes it's really hard on the ocular surface, for example, especially when you're trying to, develop an in vitro model, and sometimes the data or the information on, the state of the cell isn't really there, just because it's so difficult to sample, for example, the cornea. And so, as I mentioned, I wanted to talk mostly today around, looking at building in vitro model for contact lens, just because here that was kind of an interest that we had in trying to understand a little bit better what was happening on the ocular surface. And as you all know, there's great success with contact lens material. They improve vision, they treat vision impairment, they improve the quality of life and of people that are not seeing as well, or that have keratoconus, or that have jobs where potentially wearing glasses is much more complicated, or even in sports. And so those are great medical devices that are available. As you know, though, as many of you are engaged in biomaterials research and biomedical engineering, you also know that there's benefits, but there's also risk sometimes with medical device. And so there are complications that occurs upon lens wear, and some of them relate to, corneal ulcer or infiltrate that come within your eyes and are going to affect your vision. And also what we know, and there's been a lot of research around this, is that contact lens wearer are actually at increased risk of microbial keratitis. And again, this is an infection that can affect your vision, where you can lose some of your vision. And so one of the things we're interested as we build those models is trying to answer, questions around, why do those things happen? And is there a way, once we understand them, then you can design material better, or you can design therapies in different ways. What's also interesting, and I'm sure you're aware, is that there's a lot of opportunities with contact lens. People have started to look at them for smart contact lens so that they can, monitor glucose, for example. There's something that's been talked quite a bit around, glucose monitoring contact lens. You can also use them as a diagnostic tool, as a sensing device, and also looking at potential augmented reality. And so when you start thinking about, all the different potential for contact lens, it's also really important also to understand, or determine and design in vitro model that can actually help, prototype those lens, test them, so that we can know better in vitro if they're going to be biocompatible before they are put on the eye of an animal or on a human eye. And also what people have looked at with contact lens is also thinking about them as drug delivery contact lens, because they're right there on the eye. You could treat glaucoma, that would prevent, or reduce the use of eye drops, which aren't very efficacious, which you might have seen if you listened to Dr. Heather Sheardown talk a few months ago. We can also look at allergy treatments, also looking at, reducing infection for contact lens wear by just actually having drug delivery lenses on them. And actually what is really interesting is that recently, a few months ago, there were actually 2 drug-eluting lenses that were approved on the market. What's interesting, and which is where I kind of wanted people to think a little bit about this as we talk about, the models that we're developing and trying to understand biocompatibility. If you think about stents, for example, there's been drug delivery stents for, they're starting to be on the market and approved by the FDA around 2000. Research with drug delivery stents started around late 90s. With drug delivery contact lens, what's been really interesting is that there's been research all the way back to the 1960s, but it's taken that long to get them approved on the market. It's because the ocular surface or the ocular system seems to be much more complicated. There are other potentially challenges around it, but it just shows you how long it's taken to actually get to have those lenses on the market to actually be treating some ailment, and that's around allergy. And so what we're hoping to do as we develop in vitro model is try to see, can we help prototype, and can we help understand a little bit more potentially how biocompatibility work on the ocular surface, so that we can see and improve contact lens wear and new contact lens that will be innovative and monitoring disease on the eye, for example. And so before we go a little bit more into looking at some of the model we designed in the lab, and eventually we ended up looking at your neutrophils. Just wanted to give a very brief overview and very high level of, when we're thinking about the eye. So for us in our lab, we're really focusing on what we would call the anterior eye. And also what we're focused mostly as we're developing the test currently with contact lens material is that we actually focus on the cornea. And so when you're looking at the cornea, it's a structure that has, cells on the top. So you've got the corneal epithelial cells, you've got the stroma, and you also have at the bottom, you have the endothelium, which is actually very important as part of the hydration of the stroma. What we're looking at, though, when we're building our model, because we're interested at just trying to understand actually some of the surface interaction that's occurring, is that we're actually looking mostly or actually only at the corneal epithelium. And so what we're interested in is looking at that or designing a model that has some stratified structure, thinking about the tear film a little bit, and trying to understand is there ways where we can measure compatibility. So something beyond just being live, a dead cell or live cells. And so when it becomes interesting is that when you develop model of biocompatibility, you need to be able to also look back to see what happened in, the system that you're trying to model. And so for the eye, it's been a little bit challenging, because if you think about some of the parameters that we know or we can measure on the eye, often what you can do is that you can do impression cytology, and you can only do that on the eyelid or on the conjunctiva. You can also do tear collection. Tear collection will give you some information, or will give you information around the tear content. So you know what cytokine are there, what inflammatory mediators are present, the various protein and lipids that are present. With this impression cytology, you get a sense of the cells of the conjunctiva, but it's really hard to know what happened to the cornea, because you can't do impression cytology on the cornea. It's deemed too risky, because your cornea is very sensitive, and as a stratified structure, if you remove a layer of cells, you actually put that participant at risk of developing an infection, basically. And so when we're trying to understand, or see, what's happening to the cell on a human cornea, it's a little bit more complicated, because what you have access to is mostly microscopy. It's really hard to get a sense about, what the cells are expressing. Of course, you can have ex vivo sample eventually if the cornea is removed, but from a biocompatibility perspective, right on time, again, I was using the example of the stent. If you put a stent, or if somebody goes on hemodialysis, then you can get a blood sample and get a sense about, what is the blood material interaction with that material. With a contact lens, or for, even if you were thinking about nanogel that you may put in, or some of the releasing implant that are put in the conjunctiva, it's hard, because the only thing that really we have available mostly is looking at the eye through microscope. And often what's being used is you're using fluorescein, which basically is going to help you see, assess the ocular health. And so it's a little bit more complicated when you try to develop an in vitro model and compare with what you could see in vivo, because we often don't have that data available. And so one of the things we were interested in for us when we were looking at lens biocompatibility is that we know there is this phenomenon that's called solution-induced corneal staining, where basically, depending on the kind of lens solution combination you get, sometimes you get this, what we call the micropunctate staining, where you see those little green dots basically lighting up your cornea as you're looking with under blue light with a slit lamp microscope. And so what has been interesting is that as, we were working in collaboration with CORE in understanding this, because my goal was to understand, the in vivo situation so that I can model what we see in vivo in vitro. And so we started doing some very gentle eye wash. So we're just washing the cells in collaboration with Dr. Jones and Dr. Peterson, that was a postdoctoral fellow at this time. And so what we were interested in is trying to see, can we get a few of those punctuate dots? And as we started to do this, we could get a little bit of those corneal epithelial cells, and you can see one of them here in the middle that's stained with sodium fluorescein, and all the one around there, which are staining here with PI, so they're dead. So here you've got a cell that's stained with sodium fluorescein, and on the outside we've got dead cells. So as we were, trying to develop our model, we were also collecting data in vivo to make sure that, as we eventually design a system where we could test some of those hypotheses, we were looking at similar features between in vivo and in vitro. And so again, one of our axis of research is looking at, biocompatibility and thinking about the corneal epithelium, the tear film. We also need to think actually about the conjunctiva and eyelid, and also thinking about what's available, what's some of the microbiome that are present, some of the bacteria that are present. And so when you put a contact lens on your eye, it's going to be interacting with all those various components. And so you sometimes have to clean it. And that's why, we were interested in developing tests around this. But you also have to think about, if you're suddenly having a sensor, you're going to have your microelectronics interacting with those. And so looking at toxicity versus biocompatibility was quite important. So when we started to be in working in that area, and so when you were looking at the state of the art for in vitro testing of ocular biocompatibility around 2010, which was shortly after I started at Waterloo, if you were interested in, or what was happening is that in the research area, when people were looking at solution to clean the lens, what happened is that often, you would put a diluted solution on cells and see what happened. One of the questions what you may ask yourself is, well, how do we know what concentration of solution to use? And also, how long should we leave it on the cells to see if it's cytotoxic or even just biocompatible too? If we're looking at those two, how do we make those choices? So that was difficult in a way because we don't really know. For anything that was related to drug delivery system, what was happening is that they were the sync model, basically where you put a lens and it just releases within a certain volume, or there was also some work that was done by Dr. Barr lab looking at a microfluidic system. What you can see is that, depending on the model you're looking at, in one, there's no lens, so you're not really sure, what's happening when the solution is actually being uptaken and released. In the other side, there is no cells. And so what we got interested in is, can we develop system or in vitro models that can help, get better answers potentially around, drug delivery system and also biocompatibility around certain solution. And so one of the first things that we did is that we looked at developing a model that just had a contact lens and put that on the cells to see what would happen. And that was some work that was done with Dr. Sheardown and Dr. Jones. And so as we looked at this, and I'll just briefly walk you through, is that we're looking at, corneal epithelial cells at the bottom of the dish. Those are actually Dr. Megriff's actually corneal epithelial cells, an immortalized cell line that she had kindly given to us. And then we have a contact lens on top of it, and they were just coming from their packaging pack basically. And we were interested in saying, what's going to happen? Is there a difference between material or between solution. And so what we found during that study is actually if the cells were packaged in board buffer, we saw a change in integrin expression, alpha-3 and beta-1, which are important for cell adhesion, cell migration for a corneal epithelial cell. And so as we looked at that, we were like, okay, well, we can actually in vitro get some information about, what's happening with those glands and potentially how they may affect corneal epithelial cells. And while this was an in vitro system or model, very simple, that we didn't really develop much further, and we moved on to something else. What was interesting is that those results were actually verified by another researcher not associated with us in vivo on a rat model. And so it was kind of interesting to say, we can actually answer some of those questions with in vitro model. We all know they're limited because that never represent the full system, but there are some questions that we may then be able to answer if we can mimic some of these elements. And so as part of this, one of our interests was then, okay, well, then what happens if we do and look at drug delivery contact lens? Again, you may have seen Dr. Heather Sheardown presentation where she did talk about, the limitation around eye drops, low bioavailability, preservative, compliance. And so looking at drug delivery system to contact lens is something that's been really interesting. And so what we were interested in for this work was, when we're looking at a drug delivery system to a contact lens, can we develop a model where cells are present, and is it going to give us results that are slightly different? We weren't interested in developing a biomaterial. We were interested in developing a system to out-test biomaterial. And so what we did is that we took a normal contact lens available, commercially available. We loaded the lens with latanoprost, which is a glaucoma drug. And basically what happened with that drug is that it gets metabolized by the cells, and that's how we can treat glaucoma. And so what we were interested to see as we developed the model is that can we see a difference? Because often, as I mentioned earlier, most of the model use a no-cell model, an infinite 6 system. And so we wanted to see what would happen. We weren't surprised to see that really the release profile was terrible, because that's what everybody had reported. So at least, the model was working. So that was simple. What we did, though, after that, we were then interested in, what happened if you put cells? And if you put a monolayer or actually a stratified corneal epithelium culture. And so this is what we did, and we incubated the lens again, and then let them release through the system. And what was really interesting is that you can see what, with a release profile of latanoprost, when there's no cells, nothing is, it's not really releasing anything. As you have cells and being either stratified or a monolayer culture, there was not a lot of difference, but there was a significant difference in the release, because basically what's happening is that the drug is metabolized by the cells. And so they played a significant role here in being able to assess the drug delivery profile and efficiency of the lens. We tested the same, with drugs that are not metabolized by cells, and then it doesn't matter to have the cells in your system or not. So again, it's important as we were developing system is trying to get tools where we can actually help researcher and industry actually test and optimize the material in a way that is a little bit closer to the in vivo situation. And so for us, and the next step was actually when we're thinking about, contact lens on the eye, is that there's a constant fluid exchange. Your tears are basically replaced. Sorry, I have, but I think it's 10 microliter a minute. And so basically we wanted to be able to mimic this in vitro to see if we could get our model to be a little better. And so what we did is that we have a curved multilayer system, or a stratified culture of the corneal epitheliums, so that we can actually set our contact lens on it. And what we did is that we also, or one of my grad students, not me, but what he did is that he actually developed a tear replenishment system where we could put a curved cornea model. We would have drops of artificial tear solution. Here was keratinocyte medium to keep the cells alive too, and mimic the tear film in a way that was present. And then we also had, at the bottom, what was happening is that there was a way also for us to sample what was on the other side. And then we could see, does it actually make a difference to suddenly now, rather than having a static system, having this dynamic system where you have replenishment. And so this is kind of the end product. You've got this fairly big system that fits in an incubator. And so we were now really excited to see, what is going to happen when we start looking at, the diffusion and the transport of latanoprost on the other side, so on the basal side. And so what I'm showing you here is, latanoprost release on the basal side. So on the other side of our cell culture, the contact lens was there, so it's missing. And then what we're doing is that we're looking at drug transport and diffusion. And we've got our different lens. What you may notice is that we get lines that are the same colors. If it's a full shape versus a non-full shape, it's replenished versus non-replenished. And what you'll likely notice is that there's very little difference between having a replenishment system versus not having a replenishment system. The only time that it made a difference was with one of the contact lenses with interferon A, where we had a slightly lesser release, actually, with no replenishment, higher release, sorry. And so what was, interesting in a way was, we really complicated our life to get this tear replenishment system, but actually at the end, it really didn't make that much of a difference. And so what was an important lesson for us to learn through from there is that, yes, tear replenishment was likely important, but likely what was the most important for that in vitro model we had developed, and as we were thinking about testing or drug delivery system testing, is looking at the drug, and the fact that the cells are there was really key to actually give stronger results. And so while it's great to have a tear replenishment system because it better mimics the environment from an in vitro perspective, and some of the question we could answer with drug delivery, transport didn't really change that much, which was a little bit unexpected, I have to say. So as we looked and learned a little bit from this, what we were also interested in, as we're trying to build this model to eventually be able to look at biocompatibility with our cornea, with contact lens, and all those different things. We also wanted to see, can we reproduce corneal staining? So I mentioned to you, corneal staining is something that happened not just only with lens, but what was interesting here is that we know there are certain lens solution combinations that give rise to this, and we know that some don't. And so it was kind of not an easy way, but that was a better way to kind of look at it, because we knew what our positive control would be. We knew what their negative control would be around contact lens and what we were expecting around some of the results. And so what we did is that similarly, we used our curve stratified culture for a contact lens, just because, we're not building small model. We're trying to build a model that can actually take the entire lens, because what we were really interested in here is really trying to represent or reproduce this corneal staining pattern and effect, because especially with lens wear, often you're going to see different variation of staining depending on, if you're centered to the cornea versus the periphery. And so, we were not interested in trying to develop something that was using less cells or just a piece of material. We were really kind of interested in trying to see, can we actually represent this in real life in a way? So it's a close model. And so we're still working on it. But what I wanted to show you is some of the study we had conducted with SCORE. So, trying to understand a little better, when you see something like this, can you collect cells? And so that's what we did, is that we were able to, again, I mentioned you can't really take cells off the cornea of somebody unless, they have surgery or something. But what we could do is that we can do a gentle eye wash. So that was a way to kind of collect cells that were coming off from the cornea and actually look at them. And so when you wash the cornea, you can actually collect some corneal epithelial cells, and you can get some really interesting data and cute picture in a way, where, this is a set of corneal epithelial cells. And as you can see, they're all staining green. So they're staining with sodium fluorescein, and we have different one. We also know is that as you do a corneal eye wash like we did, is that you also get a lot of dead cells. And so that was something we were aware of and that we studied and we counted and so on. And that was part of a more clinical study that we did. And so in parallel, what we were doing in the lab is that, if I have my curve structure and I wash it and I expose them to the same lens solution combination, what can I get? And so that's where we got a little bit excited, I have to say, that we managed to see those kind of similar feature that we see on the human cornea when we've got that phenomenon. And so we can see corneal epithelial cells staining with sodium fluorescein in a way that's very similar to what is ex vivo. We also see shedding of dead cells like we saw over there. And if you actually look under a slit lamp microscope on our sample, which is a culture sample, you kind of see actually the same type of micropunctate stain that we could see around on the cornea. And so it's been an interesting mode for us, because obviously, it's not because it looks the same that it is the same. So we've been trying to look a little bit more and trying to understand, as a cell apoptotic, what kind of markers do they express, so that we try to understand a little better when we're looking at this, what does it represent? What does it tell us around the cells? And so we've been doing quite a bit of that work to try to see, can we learn something with this model to understand, what is this phenomenon that we see there. What was interesting, and that's where, as we develop all those models and we were thinking about, trying to develop a model to test contact lens and look at inflammation, because, biocompatibility and inflammation are very related in the eye, and it's really hard to know, what is a measure of a contact lens biocompatibility, or what is one for a drug delivery system, because what we have in vivo is not really, if you can do animal work for sure, but often with in vivo we mostly have pictures for the human, and so trying to see, can we develop this model that will give us a little bit more. As we've been trying different things, what happened is, as we were actually looking at doing that eye wash of the cornea, we actually came across white blood cells that were in the eye wash. And so that's something that is known, but that we got really interested in. Because basically often, when we think about, contact lens or any, not all of the medical device, but a contact lens, often you take it away, you remove it at night. Some people sleep with them, but some don't. And so most of our model were built on thinking about, the open eye environment. And so what's interesting is that when you go to sleep, something very different happened to your eye. Suddenly you don't have tear replenishment anymore, and you've got apoxia and all those different things. And so we started to get really interested in not only trying to build a model to look at, cornea during the time of the open eye, but looking at the closed eye environment. And so what we know from the closed eye environment is that there's an increase in complement activation product. We know that there's more cytokine. IL-8 is present at high concentration. We also know there's an increase in metaboloproteinase. We also know it's an hypoxic environment as your eyelids are closed. And what we know and what was known is that there was an influx of leukocyte. And that was about it. As we did our study, we were able to demonstrate that they're mainly neutrophils. But often what had happened when people were looking at leukocyte, they weren't able to collect enough to actually do any analysis on them. And so it's important when I mention neutrophils, because as you may recall from your biology or from the work that you may do, is that neutrophils are very potent inflammatory cells. And actually in the past, I would say 5 years, there's been an enormous amount of papers and research showing how important they are, not only in inflammation, but in thrombosis, in angiogenesis, in inflammation, in all kind of different disease, and even you may have heard about them with COVID-19, because they're really important in the lungs, related to lung inflammation. And so as we were looking at the closed eye environment, we found those cells and we thought, okay, well, if they're coming in the closed eye environment, we need to understand, what they do and how they behave, because it's really important from an inflammatory perspective. They must be coming for a reason. So what we did is that we actually developed a very simple eye wash system so that it could be easily done at home, and people upon awakening could actually collect cells non-invasively. And what was really interesting is that we could collect anywhere from around 50,000 cells to up to 1 million. And so what's amazing when suddenly you have that many cells is that you can actually start studying them, because before, with previous collection that had been done, they were 100, 200, and so you can't really do too much. With 500,000 cells, you can do a lot of different study, and so that's what we've done, and I'm going to present briefly some of this work. And so one of the things we showed, and that was something that was really important, is that the cells were alive. Often people had thought, well, they are neutrophil on the ocular surface, but they're likely dead or not really. Or there aren't too many. So we knew there were a lot that were coming during the closed eye environment, and we also know they were alive. And what was really interesting is that they also didn't show any sign of apoptosis. So when we were actually looking at caspase activation or annexin 5, there wasn't any of those markers present. And so they were quite they were quite active, or not active, they were quite present, but they were not ready to die, even though we collected them about 7 hours after a closed eye environment. So usually it's around 6 to 7 hours. And so that was kind of interesting. And so now that we had that many cells, what we were interested in then was, okay, we've got a lot of neutrophils on the surface of the eye. What are they? What do they express as a marker of inflammation? And so what we did is that we looked at a series of leukocyte activation marker, MAC1, which is CD11B, which is important in adhesion, and just a marker of activation in general, ICAM1, which is very important for adhesion, and so could be relevant on the ocular surface. C3A receptor, which relate to complement activation, and so around being able to bind C3A. CD66B is actually a degranulation marker, and so as you might have seen from the previous graph, there were some, or neutrophils release quite a few granules when they become activated, and so that was a marker of this activation. And then CD45 was really just for us as a way to phenotype them a little bit more from a pan-leukocyte area. And so what you can see is that our tier neutrophils are in blue, and our blood isolated PMNs are in red. And so what we did is that we took our tier neutrophils, and then we tried to activate them after we had collected them. To kind of get a sense about, what are they, what can they do? And what was striking, and you can see it in this graph, is that they really didn't do much. They really didn't activate it. So if you compare stimulated versus unstimulated tier neutrophils, they really didn't change their expression. And so that led us to believe, and that, they're non-inflammatory potentially, or they're not, activated in the same way that neutrophils that you would get from blood do, when they encounter an inflammatory stimulus, because lipopolysaccharide is, a physiological stimulus. We also did FMLP, we also did PMA. So we tried different ones, none of them work. And so they basically have this fairly quiescent phenotype in a way that was quite interesting. And so if we look at them, some pictures are sometimes good, they look a little bit different than our neutrophils. Like you can see the blood neutrophils when we try to just look, and then they started to adhere, change shape. When there was LPS put on a layer of corneal epithelial cells, they really went crazy. They were very activated. There was really nothing happening with the TRPMN. And so it really begged to us the question about, now we know this, what does it mean for ocular surface inflammation? What are their role? And if we're trying to develop an in vitro model that can help us answer some of those questions, I can't use blood neutrophils because they don't behave the same way. So how do I build a model when the cells that I'm interested in learning about are not, I can't use the regular blood that I could to run and isolate them. And so one of the things we did try to do is that, can we reproduce a tear film neutrophil or the tear neutrophil? And so we were thinking, hypoxic environment, incubation with tear film neutrophil, with tear film proteins. There's more lactoferrin, there's more lysozyme. So that's very different from what you see in the blood. Having them in presence of corneal epithelial cells, none of that work to actually reproduce some of that phenotype. So we're still working on it because we're trying to understand, what is potentially turning those cells into this phenotype, because not only is it relevant to build an in vitro model, but even from a biology perspective, an inflammatory response perspective, it could tell us a little bit more about how we can potentially turn off some of those inflammatory responses that we don't want to see, so that we can produce a similar phenotype than the tear neutrophils. And so as we continue working on them, because we've been interested in, wanting to do more work, we know they're different. And so one of the things we had to do too is, like any good scientific experiment, is that we actually needed to understand, what happened when we process them, because they're not the same as tear neutrophils. There's lots of research as blood neutrophils, sorry. There's lots of research about how you can process blood neutrophils and you can spin them down and fix them versus not fixing them. And so what we wanted to be able to do is be sure that the protocol that we have to study those cells that are eventually going to help us not only develop in vitro models, but also understand what happened potentially on the ocular surface was rigorous, so that if we centrifuge the cells, we knew we weren't going to affect them. What we did discover as we looked at that is that, fixation was had a dramatic effect on certain receptor we were measuring. Centrifugation too. So we basically know we can't centrifuge the cells more than once. If not, some of the receptors we're interested in are going to change, which, then you have to think about how you design your experiment and what you do. And we also knew that, depending on how long you incubated the cells, so this is showing, there's no legend that explained it, but it's looking at the difference in receptor expression between 0 and 4 hours. So again, those change, and being able to know how long can I use a cell before something changes was really important, as again we're trying to understand what's going on. And finally, just one last result, and then I'll close off, is as we continue to try to understand how different are they from the blood neutrophil that we see, is thinking about ocular inflammation. So we looked at some membrane receptors. That was one step, but also what we're interested in is looking at reactive oxygen species. And so we knew they were very different. Expression of membrane receptor was very different upon stimulation between tear film neutrophils and blood PMNs. So what happened with reactive oxygen species? And so what we did is that we actually look at the production over time using chemiluminescence, and we compared to what we would see with blood neutrophils. So we're looking here at the whole collection. So we know within our collection there is neutrophils, but we know there's a few lymphocytes, a few monocytes, and a few corneal epithelial cells, potentially some of them dead. And so when we compared, what we could see is that, if we try to stimulate, again, those cells that we collected after the closed eye environment, what we found with our T neutrophils is that they weren't actually quite the same. Again, not surprising potentially, but not quite the same if you activated them with FMLP, which is, again, one of those physiological activator. We could see they would have a little bit of a ROS production, which is relevant for extracellular ROS. But then what happened is the intracellular ROS production wasn't happening compared to what you would see in blood neutrophils. And so we thought, okay, well, then they're really potentially very dysfunctional because they can't properly generate ROS. What we found, though, is that if you try actually to stimulate them with PMA, which is a protein kinase activator, they're the same as blood neutrophils suddenly. And so what it's starting to suggest is that, potentially there is, is there a receptor issue around our T film leukocyte? Is there an intracellular pathway that is potentially not functioning the right way? And so those studies have started to allow us to kind of start asking questions about, how different are they, and what we're also interested in in what makes them that way. We know they are functional, like we know they can do certain things. We just don't know, how they are being turned on enough. And so that's a very interesting question for us, not only to understand inflammation in the eye, but also eventually try to be able to build a model that can help answer some of those questions and allow to do more testing around biocompatibility. And so this is kind of a little bit of a summary around some of our research and how we went from, trying to develop in vitro tasks that took a little bit more of a system approach, thinking about the various components that are present. And, there are way more. We could have conjunctival cells, and we started to do that. There's a lot of different things that you need to look at when you're starting to think about biocompatibility and the interaction between the various systems. But as you do this, what's been interesting is that it also leads to better or new understanding. And so it's been really exciting for us because we've actually generated new knowledge around those T neutrophils, which before weren't known. And, as all of us like to do, is that eventually we really want to improve biocompatibility. So we're hoping that, some of the understanding that we gain with some of the new models, or also just looking at those T neutrophils, will actually help us get there. And so that's kind of our final goal, or a long-term goal, is to eventually design or develop strategy to improve compatibility and from really wanting to, for example, reduce risk of infection or contact lens wear, for example. And so this is kind of the end of my talk. And as you can imagine, I really didn't do all that work. A lot of it is done by my students. And so some of them are only related to the ocular system, but those are some of my grad students that contributed. All the tear film work has been done by Yutong and Cameron, actually. And so the other work has been done by the in vitro model, have been done by all the other people. And also I want to thank my collaborators that have been instrumental in getting some of that research going and all the discussion. And also, as you can imagine, none of that research is easy to run without any funding. And so we've been very lucky to have support from NSERC, CORE, CBD, which is a Center for Biotechnology and Bioengineering at Waterloo, and also Waterloo Engineering and our CREATE grant that helps us actually support this research. So again, thank you very much for listening. And I'm sorry I went a little bit over time, but I hope this was interesting, and I'm looking forward to the question period.
Well, thank you very much, Professor Gorbet, for your very interesting talk. And then it was very educational. I really enjoyed your discussion about the differences between toxicity and biocompatibility. Sometimes, we think that just a simple live-dead assay is good enough for assessing the biocompatibility of, biomaterials and drugs. But it's good to go dive deep into the these biocompatibility studies, because that's a very important component of any medical device development or drug development at the preclinical stage, at least in vitro preclinical and in vivo preclinical evaluation of these kinds of technologies that are being developed. So thank you very much. So while you're taking a breath and then rest for a little bit, we'll ask questions momentarily, but I just wanted to remind everyone that our next speaker is Professor Glenn Tibbetts from Simon Fraser University. Please mark your calendars. Follow us on Twitter, TransLBME, to get most up-to-date information about the next talk, and then also the upcoming list of stellar speakers that we are putting together for the coming month. I'd like to start with questions from the audience. If you don't ask questions, then I'll take up most of the time, and you don't want that. We have a question from Professor Griffith. Very interesting closed eye model. So the question is about the type of cells that you used in your I would call immunocompetent, or the model that includes immune cells. So apart from neutrophils, have you found any other immune cells? Have you looked at dendritic cells in your model?
So thank you for the question, Dr. Griffiths. So far we've really only looked at specifically. So we know we do have corneal epithelial cells, just because it's easy to see them, I have to say, on the microscope. And we've only looked at so far monocyte, lymphocyte, and neutrophils. We haven't looked to see if we have any more. What we do know is that the population does change. So again, we're just doing an eye wash, so that's depending on how long the people have had their eyes closed, we get more or less cells, but the neutrophils have so far been the main population. But it'd be interesting to look at. We haven't thought about that. So
I think that could be a very interesting study to see what population of cells, immune cells, are present in the tear. That's a very interesting study. So let me just ask a few questions. First of all, again, I mean, developing an in vitro model is appreciated these days, especially with the goal of reducing the number of animals used towards 3 Rs, and then using less animals for drug screening and material development. So my question is about the FDA's position on these kinds of in vitro models in the area of in vitro ocular models, or in vitro models of the eyes. What is the current status? Have you been in contact with the regulatory bodies in Canada, or in the FDA, in the US, or other jurisdictions, to discuss whether these models are acceptable models for testing drugs?
Yeah, so we haven't really, because at the beginning we were more interested to see what was happening, from a model perspective, and how it affect drug release. There are definitely, so there are some in Europe anyway. There are actually some stratified system that are used for, or that talked about around toxicology evaluation of drugs. I haven't seen as much activity in that area in North America. I have to say a lot of those discussions tend to be happening much more in Europe. With the FDA, there's been some changes around how you test. For example, for lens solution and zinc, they just made some recent change. But that's been, and they're using the lens, and it's different surface areas that we do.
Okay. So the other question that is typically asked when these types of in vitro models are developed is around the validation of these models. Can you comment on how this specific model is validated, or your thoughts around the validation of the model?
Yeah, so we haven't, on that component, because of what the testing we did was, we used known contact lens material that are commercially available there. And maybe I didn't look hard enough, but we didn't see any in vivo data around some of that. Because basically, if you do it in a zinc system, nothing happens. So there was very little value. So we haven't gone yet to validating some of those models. What we've been more interested in is trying to validate some of the corneal staining model that we're doing. And again, this is where it's a little harder because there's a little less known on what happened on the ocular surface with some of this.
Okay, that's fair. So speaking of ocular surface now, so my next question is about different drug delivery systems that are being developed recently for treating ocular diseases. Can your model be used for intraocular drug delivery systems, those systems in which you inject the drug into the eyeball, and then maybe to see the drug diffusion and drug distribution within the eye, especially those drug delivery systems that target the retina and then like other.
Yeah, if it's to be injected into the aqueous humor, not really, because really, our idea is to put, it's more from a drug delivery perspective from the cornea or from the ocular surface, which it's true they are some, and that's where we've been a little bit interesting. And, there are some implant that you can put, around the eyelid or just into, I believe there's one in the conjunctiva, but I may be wrong on there. And so that's where those models that we're developing would be interesting, because that's, again, is trying to see, does it make a difference suddenly when you have the cells?
Okay, and then speaking of drug distribution, there are different parameters affecting the drug distribution within the eye, and one of them is the intraocular pressure. From what I notice in your model, you have these channels that deliver the artificial tear into the model. Have you thought about using the ports to probably model the pressure within the eye and then investigate the effect of this pressure on the drug distribution?
No, we haven't thought about it. I have to say, the complexity of the model kind of made it it's useful, but it's also very complex, and it's something that we've also learning is, where does complexity become so difficult to use, and you may not always get enough information anyway. And so we haven't really pursued this too much so far. We're still interested in using it, but we haven't started, but what could be the next stage. So that's interesting. True. I didn't think about that at all.
Yeah, because I thought about that because I thought I had a high pressure, and then I was always thinking about when I have like a eye drop, then will the drug penetrate into my eye or not? That's why I was always thinking about this.
No, and this is why it's interesting. There's so many parameters. Is that it's trying to understand what are the important one, and maybe this is more important than the tear replenishment might actually be the pressure and as it changes. I know there's been some research around it around that more from with some animal model, I believe, but I haven't seen anything else. But again, this is why it becomes interesting. It's testing some hypothesis, and you can do that a little bit easier in vitro and learn, what is important when we're testing, when we're trying to understand what we develop, if it's going to work.
Well, again, I mean, I have personal experience with. Thank God, I'm not suspicious of glaucoma. That's good, but I had a little bit of high eye pressure, and then. Again, I mean, that was scary a little bit for me. But in your model, you're using. You mentioned that you're using a tear solution. Is it possible to comment on the composition of this tear solution? Is it like a simple cell media, or you try to mimic the composition of the actual tear in your model?
Yeah, so we actually have two. So the one where we did the tear replenishment systems, this is actually medium that we use. So it's basically keratinocyte growth medium. So no serum, because that's really important not to have any. But when we worked with the tear neutrophils, we actually do our own artificial tear solution that has IgG, lysozyme, lactoferrin in it, because from an inflammatory perspective, we know that those proteins are really important. And so it's been trying to mimic that. We don't have lipids in it. And the recipe, I would say, from it is actually from Dr. Jones' lab, where they've done it before. And they do also use lipids. But for us, we've been trying to stay away just because it's a little bit more complicated. But what's been interesting is that we've been actually testing different hypotheses. And sometimes it makes a big difference to have those proteins and those enzymes present, which is interesting, because then you can start thinking about how you could potentially use this as a therapeutic strategy. So we've been kind of testing with and without and see what happened.
Well, that's fair. So my next question is about the biocompatibility or toxicities, like local toxicities and systemic toxicities. Would it be possible to combine your model with other existing models, maybe liver models or kidney models, and generate a perfusible integrated system to not only study the effect of new drugs on local toxicity, but also investigate systemic toxicities? Or have you thought about this? Do you see any value in these kinds of line of research?
Oh, there's definitely values. There's actually the name escaped me, but she actually presented at the Canadian Society for Biomaterial in May, and they've actually developed, I believe, a microfluidic platform that has actually the various organs where you can look at that systemic toxicity, which is really important. It's something that we need to consider. And again, sometimes it's important to do that with human cells, because we all know, the animal is slightly different. And that's partly why some of the work we've done has been restricted, mostly to using human cells and in vitro model, is that, for example, developing a closed eye environment model for the eye on an animal would be very difficult, because suddenly, most animals, unless you go to dogs and cats, don't close their eyes. And so if suddenly you have to suture them to actually have that environment, you're creating an inflammation. So you can't answer that question. And so that's why we focus mostly on looking at, what's happening on the eye with the human, and then trying to understand a little bit more.
I see. Yeah. Okay, that's fair. Human is, he said that he's reconnecting because his computer is updating automatically. So I encourage the participants to ask any questions that they may have in the Ask a Question box. My next question, which is the actually my last question, is that besides looking at the the activation of the immune cells, toxicity analysis, what are the others, like live death studies, apoptotic, necrotic markers? What are the other studies that you think should be added into this biocompatibility assays to further evaluate, let's say, implantable devices, let's say a drug delivery system that is going to stay on the eye for a long time, like for long-term toxicity studies?
Yeah, that's an interesting question. I think, to me, especially now that we know what happened in the closed eye environment, I think, to me, it has a huge impact on, wanting to understand a little better what's happening, and maybe thinking about how do we test for that, for biocompatibility. Because you can imagine if you have any implant, or just at the surface, because again, those are not those cells, and that's where it's really different. You're going to get neutrophils anyway in the stroma and in the tissue surrounding, but this is just part of the tear. As you close your eye, they extravasate from the blood vessel, and then they come on the ocular surface. And so if you've got, a contact lens, or if you have a corneal transplant, or if you have some kind of devices that is actually on the surface of the eye one way or another, how are the cells going to react? We know, because we've done a study like this. We actually know that if you sleep with a contact lens, and sometimes we actually are able to collect cells on the contact lens, we know that the neutrophils that we get from that lens actually have undergone prosthetic phagocytosis, for example. Their level of activation is very different from what we see from just the one that are in suspension. And so what's fascinating here is that you might know this from some of the cardiovascular area. When people do look at cardiovascular device like stents and graft, we're looking at what's flowing, so the blood, and then we're also looking at what's on the surface to understand what's happening. And some people only look on the surface, and some people only look in the bulk. What's been interesting is that we have the same situation on the eye suddenly with tears and with the ocular surface. You could look at what's on in the tears and understand what's happening there, but we also potentially know that there are cells on the lens, and if they're very activated, are they going to damage the cornea, or is it what we know that contact lens wear is you've got an increased risk of infection. Do they play a role? If those cells are very activated, are they playing a role? And so that's where, but again, we don't have an answer yet. So ideally, we would like it to be looked at. But who knows, 10 years from now, we might have said, okay, we spent 10 years looking at the cells, but really they don't do anything. I don't believe so, because they are actually, if you look at dry eye, and some people have actually used their methods to look at disease mostly, and they've seen difference. So we know they will change. And I think that's what's really exciting, is that it opens up new opportunities to understand what's happening from an inflammatory perspective.
So thank you. So your answer to my question actually led to several questions. As I promised, that was my last question. But I think I have more questions now, because the list of questions came up to my mind. One of them is about these autoimmune diseases, that some of them they so they attack different organs in the body, and one of them is the eye. So would it be possible to use your model to collect the immune cells from a patient's eye and test it on the on a personalized model? So let me just ask the first question. Can your model be personalized? Like, can we take cells from patients and then develop a model that is, reflective of an individual patient's cell or genetic profile, and then use that for drug studies? That's my first question. And the second question is about now using this personalized model to study or to screen for new drugs for treating patients who are dealing with autoimmune diseases that specifically attack or affect the eye.
Yeah, so we can. So what our model is, right now for the closed eye environment is just really trying to understand what the cells are. So we don't yet have a true model. We have a way to collect the cells, which is available for people, and it has, that's what's been really interesting, is that people have actually started to look at disease. So one of the actually Cameron Posnikoff, who is one of my former grad students, actually went to work with Dr. Nichols in the University of Alabama, and actually they started looking at dry eye, and so collecting cells from dry eye and try to understand, are they different? There is another group that has looked at another autoimmune disease, actually collecting the cells and phenotyping them again. And so as we start understanding those new population, it is definitely going to open up possibilities to actually develop models, because now we can see what they do. We can see who they are in a way. That's kind of weird, but they have a phenotype, they have a signature. And so that's going to be really helpful for us to be able to test eventually drug. We also know, they change over time. Yeah.
Okay, that's cool. So it's I see that there's a lot of potential in the in vitro model, not only for this, but maybe for other applications, and then treating different diseases that are related to the eye. So I have no more questions. But Human, if you have any questions, or if you have any questions from the audience, please.
I enjoyed the discussion, very stimulating Q&A, and so I just have one question: if you have any plan to translating these to clinics or pharmatech, this technology, or you already started doing that?
Yeah, so with the in vitro model that we have, the tier replenishment system one, for example, it's a great model for research. It's a bit too complicated for prototyping and things. So there is, for example, at Corneal, there's a company called OcuBlink, and they do develop models that are much more efficacious in a way to test some of that toxicity drug delivery system, and saying not with cells yet. We did work with them. We tested a system that's called OcuCell that will publish on that's looking at that. But yeah, currently the one that we have is much more from a research perspective. It's really cool, I have to say, because we can reproduce certain things. We need to be redeveloped so that it can be truly translated, because currently it's not. It's a little bit too complicated to operate, for example. And so,
but I mean, you can. I mean, your company can be a kind of service base.
Yes, yeah. So that's definitely something. Some of the, for us currently, and that's where, we've really gone a little bit more towards trying to understand what happened in the closed eye, for example. And so those are as also a strong potential for translation, because from a therapeutic perspective, that's actually potentially quite important. And just trying to see the difference between, closed versus open is interesting. And so from an inflammatory perspective, can we develop something that's useful useful that can help test under those conditions. Because we know, or I know based on, if I look at the response of the blood neutrophils, is that we can't use the same, or you'll get an answer, but that's going to be so different than the ocular environment that you may decide, well, we're not going to do this because inflammation is so bad. But it could be true, or it could be totally wrong, because they are a very different cell population.
Thank you so much. I don't have any question, and thank you again for accepting our invitation. I think we will meet soon in October 7th.
Yes, we are. Yeah.
So, so until then, I actually pass the mic to Mohsen to finish up, and see you soon.
Thank you. Thank you, Asad.
Sure. Thank you very much, Professor Gorbet, for your excellent talk. Thank you for accepting our invitation. I would like to also thank our participants for joining the meeting today. Don't forget that our next talk is by Professor Glenn Tippett next Wednesday. So please mark your calendars. And with that, I would like to conclude this session and then wish you a great day or night ahead of you. Take care.
Thank you. Thank you again for the invitation and the stimulated discussion. Thanks. Bye.
Bye.
Bye-bye.